Thank you very much. Thanks for coming. It's a pleasure to be here, although I must admit that I was definitely nervous to say yes to a maths seminar, because I don't really feel that that's what I do. But I've kind of used it as an opportunity to maybe potentially find some new collaborations, or kind of for you to tell me what I'm doing wrong and some of the stuff I've been doing. So I just wanted to give a little bit of a quick background, because I am so new and my group is so new here at the University of Manchester. I wanted to give a little bit of a background of kind of what we do in the broad scope of things. Cool. So yeah, these are kind of 4 areas of what we research, we study. So we are a computational lab. I do have my background in bioinformatics, but we also run a wet lab as well, a microbial lab, and we call ourselves microbiologists. So we do things like look for microbial associations in our bioinformatic data, mostly DNA sequencing data, but then we'll also grow those microbes on plates and do those experiments in the lab as well. So I won't talk about those kind of things today, or the culture enrichment that we do from sputum and various types of samples. Today I want to focus on gene-gene associations, and I'll maybe come back to some of the other bits of this a little bit towards the end. So I'd like to kind of go back to the very beginning, or what I think is, as a bioinformatician, microbial bioinformatician, was the very beginning, which was sequencing the first bacterial genome in 1995. So this is my very crude cartoon of a genome that I'm going to use throughout. So I'm just using this circle to represent H. influenzae, the first bacteria that was sequenced, and these little rectangles to represent open reading frames. Okay, so you can imagine how excited scientists must have been to have the first genome, and then perhaps how confused they might have been when they had the next few strains of the same species, because we started to see these kind of universal genes or core genes, these gray ones that are found in all the strains of the same species, but then other strain-specific genes, like some of these blue ones that are only found in that first genome that was sequenced, and other colored ones that pop up in subsequent sequenced genomes. And of course, over time, sequencing got cheaper, etc. We all know the story. So we started to have more and more different strains of H. influenzae, as well as lots of other different species, of course. So then we needed a term, a fancy term, to put all this strain level diversity together. And somebody came up with the idea of the pangenome. So the idea of, within a given taxonomic group, but usually a species, having core genes, these gray ones that we see across all individuals within that species, but also strain-specific or genes that are in some proportion of those genomes that we call accessory genes, or these strain-specific genes. And collectively, the whole thing is a pangenome. So it was already mentioned that I did some work with James McInerney as a postdoc and a fellow at the University of Nottingham. And at the time, us as kind of a larger research group, were interested in this research question of what is the genetic composition of pangenomes structured in some way? So are the genes that happen to be core, is there a reason for that? Is there some sort of structuring in the accessory genome, or is this just horizontal transfer that we see kind of willy-nilly between different strains, or because of their environment, something like that? And myself, others in the group, others outside of the group, took all these different types of approaches to answer this question. So for example, James McInerney and Alan Bevan have a new tool out using machine learning to come to some of these conclusions. Maria Rosa Domingo Sanchez did a lot of work with effective population size, as did James McInerney and Alan McNally, kind of the first paper within the group that got this started. And you'll see some mathematical modeling, but that's not what I did. So I was interested in looking at the relationship between what we call the lineage independent genes, which I'll define in a second, but essentially accessory genes within the pangenome. So this is work that's published now, but I want to go through it a little bit in the first half of this talk, just to kind of lay the groundwork for some other work that I'd like to show you that's currently ongoing. So to look at the relationships between lineage independent genes, we first have to define them. So within the accessory genes, so genes that we see in some but not all strains within a pangenome, The lineage independent genes are ones that we see that aren't in a specific lineage, right? So ones like this pink gene that we see kind of across the phylogeny, species phylogeny. And that's in comparison to something that we would call lineage restricted, like this blue gene that we just see within one clade. So I was less interested in having these lineage restricted genes as part of our analysis because the most parsimonious example or explanation would be that that blue gene was in the most recent common ancestor in that clade, and then it just moved through vertical evolution. So I was more interested in ones that looked like they could have been acquired horizontally in different aspects of the phylogeny, or that they had just been maintained over time from some sort of vertical evolution. So these lineage independent genes we wanted to know if this variability was structured. So they're an important aspect. As you'll see, there is quite a few of these genes within pangenomes. And we wanted to ask if they were under evolutionary pressure. Were they under any selective pressure to be in the certain strains that they're in? Or was this just, were they just present by chance? So this is kind of embarrassing to show at a math-based seminar, but this was kind of the idea, the basic algorithm of what we did. So we'd have, for example, that pink gene that was present, so lineage independent gene present in some strains, but not all. And we'd just ask the question when we compared it to other genes, if those two genes were present more than we would expect by chance, okay? And if that was the case, we would build them into our network as nodes and draw an edge between them. So that's the extent of my mathematical knowledge, right? So we kind of built this up and did this for all the lineage independent genes within a given pangenome and would start to build these networks of co-occurrence relationships. This is what I wanted to do at the time, but there was no piece of software to do it. So myself and Martin Russellowicz, who's a really, really talented postdoc with James McInerney as well here in Manchester, we together wrote this tool called Coinfinder that can look for these relationships. So once we had the tool, we applied it to a few different pangenomes, but this is the one that I'm going to talk about today. And looking across actually the genus in this case, not just the species of Pseudomonas. So we had 209 genomes that were publicly available and just under 100,000 homologous gene clusters or genes across these genomes. So to give you an idea of kind of what this looks like, when we look across the genomes, we can look at the distribution of the different genes and how frequent they were in the data set. So we have some number of genes that were found in every single genome. And then we have a lot of other genes that are quite rare, just found in one or two strains. And then this kind of almost U-shaped distribution that is kind of known in the pangenome literature. We see a little bit of a spike in the middle, probably due to an overrepresentation of one particular species, Pseudomonas aeruginosa, because it's of clinical importance. So in our analysis and using Coinfinder and seeing if it would do anything of interest, we focused on our abundant accessory genes. So we're excluding those genes that are found in every single genome because they're core, so they're not interesting for answering our research question. But we also can't say anything statistically about genes that co-occur in every single genome or potentially co-occur in every single genome. And then similarly, we don't look at anything that's rare. So we just don't have the statistical power if we're only seeing a gene in one or two or three genomes in the data set. So we focused on the abundant accessory genes and then used an algorithm that was previously published to determine which of those are lineage independent. So just looking across the phylogeny and removing any of those genes that are found in a particular clade. So when we applied Coinfinder to this set of genes, this is the network that we obtain. So this is similar to that cartoony network I showed you a few slides ago, except applied to this data set. So the colors might be a little bit hard to see if you're in the room. But if we kind of walk through this, we have just over 6,000 gene clusters, so 6,000 nodes or dots within our network. And these are connected by just under 300,000 edges or links. And that means that we have just under 3,000 co-occurring gene relationships. And then we can orient these into connected components, which are the colors in the network. So this is kind of sets of genes that not necessarily every single gene co-occurs with each other, but there's this set of relationships within them to get these connected components. So in terms of the biology and kind of digging into what this network means, besides being this fancy thing that you can put on a slide, this is actually the really ugly figure. That was the thing that I was the most excited about in this analysis. Because what it shows is that the majority of the abundant accessory genes, so the genes that we looked at initially in our analysis, the majority of them formed at least one co-occurrence relationship with another abundant accessory lineage-independent gene. So we had about 8,000 of these genes that we started with, and almost 87% of those formed at least one relationship. So that starts to potentially answer our question of perhaps there are evolutionary pressures. We're tending to see genes together in our analysis. But what's next? So we have to dig into what kind of relationships we're finding. So the next thing we asked was, do these genes share functionality? So we had a randomly permuted data set of gene pairs from the same starting data set that we used to compare everything throughout the next few figures and analyses. So annoyingly, we kind of change our terminology here. So associating and co-occurring are synonyms. But what we found is that genes that co-occur or associate with each other are more likely to share overlapping Go annotations, so similar functionalities, compared to two genes from that starting data set that are just randomly pulled together. So what does that mean in real life? So this is an example of some transporter genes. So if we pull out that Go functionality of genes that help in transport in Pseudomonas, this is pulling from that larger starting network. This is the network that we get. So we have these, I've numbered them here, but we have these sets of components. Some of them are really small, just including maybe 2 genes or 3 genes. And then we have larger sets of genes that are co-occurring just under this heading of transporter function. And in terms of the heat map of all of our strains here in the phylogeny, this is the presence absence of each of those genes within the same numbered connected components that we have. So you can see some, for example, 7 and 2, or maybe arguably have a little bit of a phylogenetic signal. They're not fully kind of lineage independent, although they pass the cutoff. Whereas we have other clusters that are definitely widely distributed across the phylogeny. Okay. So next we asked whether these co-occurring genes were co-expressed. So if we think that they're potentially together in certain strains of these genomes because they have similar functions, perhaps they're then expressed at the same time. So using publicly available RNA-seq datasets, so each of these dots represent different experiments from public data. We just had a look to see whether genes that co-occurred or associated with each other were more likely to be expressed at the same time in a given experiment compared to these randomly paired abundant accessory genes, which is what we see in this data. And similarly, we see that co-occurring proteins as the products of these genes are more likely to interact with each other as well. So we had a lot less data here to start with. You'll see the numbers are a lot lower because we're relying on the string database. So we're relying on really these genes or the protein products of these genes to have been studied and put into this database. And because a lot of the genes that we're looking at are hypothetical or low abundance or not in the fancy clinical strains, we just don't have any data for that. But when we did, and when we could look at what's in string, we were more likely to find the protein products of our genes that we found to co-occur to have that an annotation of protein interaction within string compared to our random paired genes. And just to go back to that idea of hypothetical proteins, so I can't remember the exact proportion, but a very large number of the genes that co-occurred were just labeled as hypothetical. as I think most of us know as bioinformaticians, that this is such a big issue in our data sets. So just as one example, so there's this AMP-C gene, which is associated with antimicrobial resistance, beta-lactamase activity. And again, just pulling from that larger network, just pulling out that gene and anything it's associated with. So you have the AMP-C gene in the middle here in red. And then I've colored all of the genes that it has a significant co-occurrence relationship with as either yellow if we have some idea of what that gene is. The ones that actually have gene names, I've labeled on the network. But everything in gray is just listed as hypothetical. So we don't know really anything about these genes. And again, the heat map here of just presence and absence. This is our AMP-C gene that's highlighted with the kind of background rectangle, I guess. And you can see this set of genes is not super common across all the genomes, but their co-occurrence relationship is quite well conserved. They're really spread out. They're not just all next to each other in one operon. And we have no clue what the vast majority of these do. So I actually pitched this, I don't know, 3 or 4 or 5 months ago at an AMR workshop as, you know, potentially a way to go look at things like, what are these genes doing and how are they contributing to AMP-C biology? So perhaps kind of this could be a way of building hypotheses as to potentially what these hypothetical genes might be doing if we look at what they co-occur with. Okay, so conclusion so far. So this is all published. This was published a few years ago. Hopefully I've shown you that these co-occurring genes that we can find with Coinfinder have potentially some sort of biological function. We have evidence that they're under a selective pressure, that they share functionality, are co-expressed, and when we have the data, look like they produce proteins that co-occur with each other. So this is published. This tool has been used by us and others on different types of data sets.